Hi everyone, my name is Vayu Maini Rekdal. I have the great pleasure to be one of the winners of the Birnstiel Award this year. And I'm very grateful to the committee for awarding me this prize and to all of those who made this possible in the first place. So today I'll share a little bit about my thesis research that I did at Harvard University in the lab of Emily Balskus. And there I studied microbes. We live in a microbial world. Microbes occupy virtually every corner of the planet, from the bottom of the ocean to the soil, to food that we like, fermented foods, and even our own bodies. And in all of these places, it's really the microbial chemistry that allows the microbes to live in such diverse places. And this chemistry also allows the microbes to profoundly impact the environment around them. And we can see this impact inside our very own bodies. We are home to trillions of microbes living mostly in our gastrointestinal tract, in our large intestine. And these microbes have been associated with human health and disease. We know that they're very important for our bodies, for how it functions. And we know that the way that they communicate and interact with us is through the language of chemistry. The microbes have this vast metabolic potential. They encode for 150 times more genes than the human genome, and that gives them the ability to produce and consume a lot of different molecules, and that impacts us directly. Now the challenge is that even though we know that the chemistry is really important, we don't really understand how it works in molecular detail. So gut microbial chemistry is poorly understood, and this is kind of the general area that I try to tackle during my PhD. So one area that this is evidence is in the context of xenobiotic metabolism. So xenobiotics are foreign ingested substances, such as drugs or dietary compounds or food that we ingest. And when microbes encounter these molecules that we ingest, they can modify their chemical structures. They can actually degrade these molecules. And in the process, they can change how these molecules interact with our body. And this has consequences for nutrition. You can imagine if you eat something, the microbes are modifying it. That's actually going to have consequences for how that food will impact you. The same thing goes for pharmacology. So if you take a medication or a drug, microbes modifying that drug will really change how it works inside the body. So we know that xenobiotic metabolism is really important and can really impact us, but we do not really understand often who is responsible for this metabolism and how. How does this work? This is the central question of my PhD research, and I'm excited about answering this question for a few reasons. First of all, if we can answer this question, we can better predict how foods or drugs will impact people, so we could design better medicines and better foods. In addition to that, we could manipulate specific pathways. So you can imagine we could block and inhibit pathways that are not beneficial, or we could promote the presence of pathways that are good for us. And finally, by studying this question, we could really identify new chemistry and new biology. The microbes in our gut are this really complex biological system that we don't understand much about. So there's a lot of fundamental insight to be gained. So I started answering this question with a specific drug called L-dopa. So L-dopa you see here. And we know that this drug L-dopa is converted by microbes into a product called M-tyramine. And a lot of work from back in the day had established that this goes through a two-step pathway. The first step here is called the decarboxylation reaction, and the second step is called the dehydroxylation reaction. And these two steps is really what I tackle in my PhD. Which one out of the many trillions of organisms are responsible for this chemistry? And then inside that organism, what is the enzyme? So first we tackle this question of decarboxylation. And before I get into this, I want to contextualize why I thought this was such an important problem to study. So L-dopa is the main treatment for Parkinson's disease. Parkinson's disease is a neurodegenerative disorder that's marked by low dopamine in the brain. And so to replace that brain dopamine, people take L-dopa. They take it by mouth, it goes into the brain, and there a human enzyme called AADC converts L-dopa into dopamine. So L-dopa replenishes dopamine. The challenge with L-dopa is that not a lot of it reaches the brain. Quite a big fraction of L-dopa is actually metabolized outside of the brain, in the gut. And when this happens, the dopamine that's produced outside of the brain cannot reach the brain. So that's no good. And this can be done by the human enzyme outside of the brain, but also we know that gut bacteria can do this reaction. So we were thinking if we can identify how gut bacteria break down L-dopa, we might actually be able to make Parkinson's disease treatment more effective. So we first tackled this question: how do gut microbes break down L-dopa? To make a long story short, we identified that the organism Enterococcus faecalis uses an enzyme called TYR-DC, or a tyrosine decarboxylase, to break down L-dopa into dopamine. And we had identified this reaction, this enzyme. We developed a molecule called AFMT that could block this reaction from happening. And we're very excited about these findings because we think that they have implications for potentially for Parkinson's disease. And we're excited for the field to kind of build on these findings and explore these in a more clinical setting. And our paper was published earlier in 2019, and there was a report that was published at the same time that found very similar things, which we're excited about. Now, the major focus on my PhD was the second step, the dehydroxylation of dopamine into M-tyramine that you look at here. And you may wonder, why is this even interesting? Why would we care to study the second part of the reaction? Well, there's a few reasons. The first reason is that we think that the dopamine dehydroxylation reaction that you see here could be involved in the side effects of L-dopa. It's also very interesting to study because dopamine is a human neurotransmitter that's already available and produced in the human body. And then finally, this reaction on its own is very chemically challenging, and we don't really know how it works on a molecular level. And so we could really learn something new about chemistry. So we were wondering, what organism does this reaction, and then what enzyme is responsible for this reaction? So let me first tell you about the organism. That's what we're going to focus on first. So we first took a library of different gut microbes, 20 or so that we had in the laboratory. We incubated them with dopamine and we looked at whether they could do this reaction. None of our 20 different gut microbes that we tested could actually do this. So we had to turn to an alternative strategy. We recognized that on a chemical level, the dopamine dehydroxylation reaction is similar to something that occurs in the soil. This reaction is called dehalogenation. It's the removal of a similar electronegative atom from an aromatic ring. And in the soil, we know that this reaction, this chemistry, helps microbes grow a lot better. So they actually can use this as an electron acceptor and they get a boost in growth. And we were wondering, because the chemistry is similar, maybe the biological effects of these reactions could be also similar, such that in the human gut, dopamine maybe could promote microbial growth in a similar way. So this was our hypothesis, that dopamine could promote gut microbial growth. And so we used this to isolate a dopamine dehydroxylating bacterium from the gut. So we took a human microbial community, aka a human stool sample. We put it into a medium, a growth medium, that was lacking in electron acceptors. And we added dopamine here as the main electron acceptor. And the idea here is that those organisms that can use the dopamine to grow would get a benefit over those that cannot. And then over time, we can isolate the microbes that can use the dopamine because they will have taken over the community. And so we took this approach and we were able to identify the first ever dopamine dehydroxylating strain. This organism is called Eggerthella lenta, or E. lenta for short. So we found that E. lenta can dehydroxylate dopamine into M-tyramine. And now we turn our attention to the enzyme. What enzyme in E. lenta would be responsible for this interesting reaction? Again, we took inspiration from what we knew about happening in the soil. There was an enzyme called 4-HBCR, reported a while ago, that performed chemistry that was quite similar to the chemistry that we were looking at. The dehydroxylation of an aromatic ring. You can see that this para-hydroxyl group is being removed from 4-hydroxybenzyl-CoA, and we thought, well, these reactions look quite similar. So let's take the sequence of 4-HBCR from a soil microbe, we search in the E-lenta genome, and then we should find our enzyme. But we have no hits. We were actually very disappointed because our hypothesis was not correct. There was nothing similar in the E-lenta genome. So we had to find an alternative strategy to find the enzyme. Here we turn to what's called RNA sequencing. This is a strategy that had been used previously to identify enzymes in E-lenta. So the way that this experiment worked is that we grew E-lenta with and without dopamine. And the idea is that dopamine would turn on the gene that is responsible for breaking down dopamine. So that dopamine would be an inducible kind of activity. So we grew these and we then compared the genes and we did RNA sequencing, and we found that only a very few genes were upregulated. This is the kind of a plot that shows the position in the E-lenta genome on the x-axis, and on the y-axis you see how much was a gene upregulated with dopamine relative to without dopamine. And you can see there's a few genes that are contained within this kind of green box, and one of them really was striking as a potential candidate that you see here. It was highly upregulated, actually the most highly upregulated gene in the whole data set. So we thought this is a promising candidate, and we spent a lot of time trying to confirm the activity of this enzyme. And that was a very challenging task, and we tried many different strategies. Eventually, we kind of did something very old school, where we grew up E-lenta, we fractionated the lysate, and we did activity-based purification, eventually ending up with a fraction that had the enzyme of interest. And we were correct that this enzyme that we identified through RNA sequencing could convert dopamine into M-tyramine. Very, very excited about this finding. And we call that enzyme DADH, or the dopamine dehydroxylase. And that kind of identified this mystery of the second part of the reaction. The E-lenta uses the dopamine dehydroxylase enzyme to convert dopamine into M-tyramine. And with that, we're able to identify this kind of long-standing question in the field of how is L-dopa metabolized by gut microbes. And we found that the first organism and enzyme, the second organism and enzyme, and we also found a molecule that could block this reaction from happening. And I just want to kind of sort of share a little bit where this led us. It turns out that this kind of study of the second part of the reaction, the dopamine dehydroxylation reaction, sort of led us on this whole journey that I will not talk about in detail today. But what I want to share is that this dopamine dehydroxylating enzyme is a founding member of a whole new enzyme family that is widely distributed in the environment. And this enzyme family will take molecules such as dopamine and dehydroxylate them. And this includes molecules that are present that we eat in our food. And so we're very excited to identify that much broader enzyme family that is relevant to human health, but also is present in the soil, in microbes that live in water, in bacteria and archaea. And we're excited to see kind of what people might find exploring these enzymes in more detail. So I want to go back to where I started. I started asking this question, who is responsible and how does this work when gut microbes break down xenobiotics? We know that this is important, but we often don't understand how this works in detail. And I hope that I've convinced you today that this is a worthy subject of study. For example, it has implications for human health. You can imagine that we identify microbes that break down drugs, we may be able to stop that metabolism from happening. We could inhibit it. And we have demonstrated kind of that as a proof of principle in our study of L-dopa, the treatment of Parkinson's disease. So with that, I just want to thank all the people who made this possible, my family, my partner, my mentors, of course the committee for awarding me this prize, and also fantastic lab members in the lab of Emily Balskus, my professor Emily Balskus, who really provided the resources for me to thrive and develop as a scientist, the team that I worked with towards the end of my PhD, collaborators, and my dissertation advisory committee, as well as my funding. And particularly, I want to be shout out, kind of very grateful for the Gilliam Fellowship from HHMI. And again, thank you so much.